Well, I think there is some overlap with the previous presentations, and I want to discuss a little more on regen part. Well, as we well heard this throughout the whole morning, well, we know that mountain pine beetle attack area is increasing, and somewhat it is exceeding the salvage logging capability. And we have very limited time because there is some treefall dangers and other dangers associated with this killed stand. So we have only limited time to do anything for site preparation. But this lodgepole pine regeneration is, highly required organic layer removal because it has very small seed and it has very little capability to survive. As you saw at the end McIntosh's presentation, if you don't do any site prep, usually those seedlings just doesn't survive too long. So the organic layer removal is highly required to regenerate those killed stands. However, those stands usually have very thick organic layer, so we need to do either mechanical treatment or prescribed burning. And we do know that the mechanical treatment is quite effective and it has been applied in many areas. And we know that we can regenerate a stand using mechanical treatment. However, to apply it in a larger area is quite expensive and it's labor intensive, which means that considering the large area of mountain beetle attack, we may not be able to apply it under time restriction. So we are thinking, can we apply burning treatment to this area? If you can burn it with under good control, it might be cost effective because all we need to is ignite and nature will take care of it. And however, it's somewhat unproven. There are some debate that is it really working or not, because in that stand we may face high fire risk or fire danger. So if it is uncontrollable, we may get into trouble. We do know that fire behavior will change after mountain pine beetle attack, such as it will change the available field loading, and it also changes the field arrangement, because a lot more field will move down from canopy to the surface. So there will be different field arrangement. Moreover, we expect that field moisture dynamics will be also influenced. Some people argue that crown fire possibility may increase, but it's highly debatable and we do not have much of solid idea or solid information to support in either way. So we really need to understand, we really increase the intensity of fire or not. And it will change by the time since outbreak, so it's also highly debatable in cases. So can we really put prescribed burning for surface field reduction? If we can do it and if you can apply it in large area, I think we have a good solution for regeneration. So that's where our study idea started. So we have two main questions. The first one is, are those treatments actually effective on regeneration? So we want to see if these treatments will influence on seed source, so how much seed is coming from the canopy or canopy, and how are they influencing the seed germination, and how are they influencing seed growth. So this is the regeneration side of the project. And the other side is, if you want to actually apply this prescribed burning, we need to understand the fire behavior under this disturbed condition. So when we are applying fire, what will be the fuel consumption? Because even we can actually burn it, if we do not consume enough amount of fuel, it may not be really effective. So we really need to know what is the fuel consumption and what is the fire residence time and what is the rate of spread. So those basic information need to be gathered, which is not known yet. Moreover, we need to understand field moisture dynamics. So when and what is a proper window to do the prescribed burn. And I'll explain a little later but, about this a little more. So we have two study sites. One is Jackfish site. So this was attacked about 5 years ago. So this is entering into the gray stage. So this is a natural attack area. And we have Horse Creek, and this is artificially killed. We injected herbicide and we killed the stand. It's a little outdated stand, and right now it's almost all red and it's fully killed. We aim 100% kill, and we are quite confident that we achieved the goal. So we applied herbicide next spring, so it has been about a year now. And for the Jackfish site, we have 13 plots, and we will have two levels of burn, which is high intensity and low intensity burn. And in fact, probably it's more proper to say high severity burning, because we are trying to burn a little more, the longer fire residence time we are hoping to get. And there will be a mechanical treatment, and there will be 4 reps for each treatment. And slightly different layout, but basically the treatment concept will be identical. So for the first question, regeneration part, as I mentioned before, we will evaluate what will be the seed rain and how those treatments will influence seed rain amount. And seedling, we will do some seeding experiment to see the germination study. And we will also count seedling density. And the growth part, we also measure how much biomass gained for 2 growing seasons. And we also understand or evaluate the nutrient condition of under this treatment. So we have built a seed trap, and we will put 5 seed traps in each plot. And we will collect it 1 week after fire, and we will collect continuously 1 month interval. And for germinations, we will see how much natural germination we are observing. So using transects, so we will run 2 transects per plot. One will run from northwest to northeast to southwest, and another one will run from southeast to northwest. So there will be 2 transects running through each plot. And for every 5-meter interval, we will set up a 1.78 meter radius circular plot to count how many germination is occurring. And we will record it 1 week after treatment and 1 month interval afterward. Moreover, we will actually do some seeding experiment, because what we want to know is how much seed is coming, because one alternative source of seeding is in the ground, because there are some studies showing that the Thornton's cones, the closed cone buried in the soil, may stay there with the active seed. So they may germinate after the seedling, after the treatment. So we want to see that how much are coming from those buried seed and seed source and everything else. So this natural germination study will tell us in natural condition how much seed will germinate after this treatment. So another one we want to see is how those treatments are actually effective. To know the effectiveness of those treatments, we will do seeding experiment. So we will actually put seed in 12 subplots of each plot. So we will have one basically seed capsule, so with seed cap with 10 seed, and we will put another the cone without seeding. So by doing so, we will be able to count what is the percent germination we are observing after this treatment. So after these several first month of seeding counting and germination counting, we will try to see what is the seedling growth. So after first growing season, so end of first growing season, second growing season, we will count how many seedlings have established and survived in each plant. So we will run, so we will run the same transact as we did for our seedling counting. And at the end of each growing season, we will have our same circular plot, and we will count how many seedlings actually survived after one growing season. And for the root column diameter and for those seedling height, we will have 30 by 30 small rectangle square plot at the center to see that how actually not only they are survived, but also how well they are growing. Moreover, at the end of second growing season, we will harvest those trees and measure how much biomass gain those seedlings had. So using this information, we will compare the above ground net primary production of these treatments. Also, we will do some nutrient analysis, and it will indicate to us how well those treatments were effective in nutrient cycling wise. In nutrient cycling wise, we will also do several more additional studies. The first one is the soil nutrient. So we will collect soil samples before the experiment, and 2 years after the experiment, we also collect another soil sample to see the nutrient condition difference. We are targeting to measure soil pH and exchangeable nitrogen, phosphorus, calcium, and magnesium, and potassium. In the forest ecosystem, usually nitrogen is the limiting factor of the tree growth. So nitrogen mineralization is a really important factor to determine the tree growth and productivity, sometimes the survival shifts. It is very hard to measure precise amount of nitrogen mineralization, and each method has its strength and weakness. And we believe that in this condition, which will have different moisture content, resin capsule would be a good method to apply. So we will put 6 resin capsules per plot, and we will measure nitrogen mineralization. And we are hoping to compare the pre-treatment condition and 1 month after treatment, because usually in this condition, treatment increase soil temperature and sometimes increase the nitrogen flux. So we usually see high nitrogen mineralization within 1 month after treatment. So we will see 1 month after treatment. And afterward, we will try to capture a little longer period, such as over the winter, the nitrogen mineralization, and growing season nitrogen mineralization. Another way to see how nutrient is cycling is litter decomposition. So we are planning to do litter bag study. So we will install litter bag after the treatment, and in each given time interval, we will actually take the litter bag out, and we will measure how much biomass loss has occurred. Also, we will see how much of lignin content was there, and moreover, we will measure nitrogen and phosphorus content. So through this method, we will see how the decomposition occurred. According to previous studies, it is shown that preserve burning has influence on soil microbial community, which is indicating that under burn condition, we will see slow decomposition rate. However, it is not fully understood that how those fire will influence soil microbial community. So I think it will be an interesting study to see how long the nutrient will stay in the system. So our second question is, as I indicated, we will try to see and try to evaluate fire behavior during the treatment. So the first one we want to see is it really effective? If we are actually burning it, and how effective is it? And second one is, if it's effective, what is the proper window we can do? Because we don't want to apply the prescribed fire when it's too wet or when it's too dry. Because when it's too wet, we may not be able to burn enough depth, so it may not be effective. If it's too dry, we may burn it too severe, intense, or initiate crown fire. So it's something we really need to know if we want to adapt and apply prescribed burning for regeneration or to regenerate lodgepole pine. Another possibility is, as I mentioned, there is a possibility that those prescribed burning can initiate crown fire. So although we may not be able to answer the whole questions, but we kind of have a glance on it. So one thing we will measure is the depth of burn. Depth of burn has many implications, but at least in this project, it has very direct indication of effectiveness of burn treatment. Because if we cannot consume enough amount of organic layer, this treatment may not be applicable to the lodgepole regeneration. So this is a duct pin, and we will install this on 16 points. This is 50 meters each. So we will have 16 duct pins in each location, and we will measure how well this fire is consuming the organic layer. Yeah. Moreover, another thing we do not know is how those fire on surface, which in surface fire usually goes about 600 to 700 degrees during the fire combustion. And we do not know how those heat transferred into the canopy. Because if it actually hit to the transfer canopy too hot, that means we may cook the seed inside of serotinous cone. And those seeds in the cone may not be viable. So, and this temperature transfer is not fully understood. So we will, using some balloon and some fishing guideline, we will install some data logger with temperature probe, the data logger, in the middle of canopy. So by installing those instruments, we will hope that we understand how much temperature was actually transferred into the canopy. And this is kind of some initial study, so we may not be able to answer all the questions, but we hope that this will give us some gain in our heat transfer models in fire propagation. When Beetle attack and it goes red to gray stage, we do know that it generally decreases transpiration. However, evaporation might be almost identical because needle usually stay there quite several years. I believe that in ants study, little loss was less than 5% for first few years.
I only looked 1 year out.
Yeah, 1 year less than 5%. That's what I heard. So when you are losing only less than 5%, usually the light penetration is almost identical, and evaporation will be almost equal. While you are losing some interception capability in the canopy. As a consequence, we may have increased field moisture, or we may decrease available field load, but it has not been fully quantified. This is something we hope we can achieve through this study. Another one is we are trying to see if we can actually apply or we can modify the fire weather index. Fire weather index is widely used and very effective indices we are using to predict fire danger, and it is also used to apply prescribed burn. Because this is indicating how much field moisture we are having. So there are several studies showing that, and this was basically developed from given field moisture condition of the forest. And however, this index is developed for closed canopy and living forest. We do not fully understand that how this index is applicable to killed or dead stand. So we are hoping to develop or modify our current FWI index and see that how each index is actually representing what moisture content of soil, the field at each depth. So we will measure little layer and live bed moss and fermented layer and humus layer and soil. We will actually collect it, and using the laboratory gravity moisture technique, we will actually measure what is actual field moisture in this depth. And we will relate it to the FWI index, and we will try to calculate or to make a general model which allow us to predict field moisture condition at each fire weather index. So for 8 plots, so which is for control and for disturbed control, and we will do 4 reps per time, and we will measure more than 20 times along the varying FWI indexes. So this is kind of the previous report of somebody else. I forgot to put a citation, sorry about that. So you can see that this is the DMC, depth moisture content, and depending on the forest types, and they tend to show different patterns. So this is the DMC number and actual moisture content. And we are hoping to develop something very similar for this mountain pine beetle killed simulated site. There are 4 factors or 4 major factors influencing crown fire initiation. And they are canopy base height, canopy bulk density, and surface fire intensity, and foliar moisture content. And all these 4 factors are very important to determine crown fire initiation. And this prescribed burn usually consume the surface fuel, and they tend to decrease surface fire intensity just because there is less fuel. Moreover, it will change several other characteristics. However, in this mountain pine beetle, we are expecting to have low foliar field moisture because it's killed, and they usually controlled by the direct moisture exchange in the atmosphere. However, the condition of this foliar moisture is not understood. So we are trying to have a pilot study and to see that what is the moisture content at the bottom of the canopy, because this is a location that will start a fire, because fire usually starts from the surface to the crown. So for 5 dominant trees per plot, we will measure the foliar moisture kind of monthly level. So we try to see the pilot study. So out of this study, we have several expectations for management. So we try to evaluate if controlled burn can be an effective and inexpensive generation tool or not. Also, we want to compare the effectiveness of burn and the mechanical treatment on regeneration. And we also want to evaluate the seed bed preparation by burn. And we also want to know if it is really effective, what is proper or best window for the prescribed burn. And if also this is effective, we want to know that how much field is reduced by those treatments, especially burn. And we want to understand how those mountain pine beetle is influencing crown fire initiation and other fire hazard and fire danger. We are hoping to have this project started last year, but last year we could not really get it going because we call it as a Goldilocks syndrome, and sometimes it's too hot to burn, sometimes it's too cold to burn. So last year was too hot, as you know. We lost a little over 900,000 hectares of forest, and when that happened, we don't have much of, we don't really want to burn our forest too. So we could not burn it last year, and we hope that this year is not too cold nor too hot, so we can actually burn our forest and get done our research. And we have a lot of help from here and there, and we have many project members, including Vic Lieffers and Derek, and there's Contilier and Dave, and we have 3 master students working on it, and we have high help, a lot of help from the app innovation and the warehouser.